We're going to be talking about glycolysis. So like many of these other sets, we'll have an introduction video and then get into more details. So glycolysis is going to be the chemical basis, and we're going to be talking about also the logic behind glycolysis. This is going to be the central pathway of metabolism. So before we were looking at those giant metabolism maps, there was this giant pathway in the middle that was highlighted, and that was glycolysis. So now it's time to look into it in more detail and look at how this works. This is going to be looking at how we break down just the glucose. So remember that there's a lot of different dietary polysaccharides, starch and glycogen specifically, cellulose if you're a cow, or small bacteria. Those are polymers that are just of glucose. So there was just a whole different field that were just glucans that are polymers of just glucose. So the ability to break down glucose is really useful. We'll talk about glycogen later because that one's also important and useful. So as far as the metabolic fate of glucose, we're going to talk about how that degradation is going to give us energy and how it's going to give us some energy conservation because it's also occasionally going to require some energy to go in there. So we'll look at that as well. Glycolysis literally means splitting sugar. So the lysis part of that is the split. Glyco for sugar is, as you would have imagined, since we are breaking something down, a catabolic process. This is also going to be an anaerobic process. As we look at the entire process of not just breaking down glucose, but we'll also be later continuing on into the citric acid cycle and then the electron transport chain, we'll eventually get into things that do require oxygen, but just glycolysis, no oxygen required. So it's actually evolutionarily predates the appearance of oxygen in the atmosphere. It does actually have another name, so this is one of the things that luckily did get renamed, but you may also hear this called the embedded Meyerhof or Warburg pathway. This basically all cells carry out glycolysis. Glucose is that essential and that commonly used in our world. It's got 10 different reactions. We'll be looking at each one of these in turn. Not in individual videos, but for some of them will actually be individual because some of these will actually also be regulated steps. And so those will be going into more detail on. You're going to see two main phases. We'll look more closely at that in just a minute. Your products out of glycolysis are just going to be pyruvate, ATP, and NADH. So really only 3 products to concern yourself with, but lots of steps to get there. And there's 3 possible fates for pyruvate. So looking at those 2 main phases, and again, we're going to go into all of these steps in more detail. So this is just trying to get the bird's-eyed view. So phase 1 is going to start with glucose. That's going to be phosphorylated. It's also going to go through an isomerization. It's going to be phosphorylated again, and then it's going to be split into 2 different molecules. And then we're also going to have an isomerase so that even though we wind up with 2 different molecules, we actually are only going to worry about one of them. So then when you see the second phase, suddenly you see that at the bottom of phase 1, we have dihydroxyacetone phosphate and we have glyceraldehyde 3-phosphate. And then suddenly when we look at phase 2, all you see is glyceraldehyde 3-phosphate. Well, that's because at the end of phase 1, whatever amount of dihydroxyacetone phosphate we have undergoes another reaction to get that dihydroxyacetone phosphate to a molecule of glyceraldehyde 3-phosphate. So then when we begin phase 2, actually everything in phase 2 is duplicated. So when you look at this, you actually see these little 2s all throughout everything. And that's because you actually do have 2 molecules of glyceraldehyde 3-phosphate, which means to do that first reaction, you would need 2 molecules of inorganic phosphate, you would need 2 molecules of NADH+, and you would form 2 molecules of NADH as we go along. So that's one of the only strange things in phase 2 is everything's actually happening twice. Everything's doubled because of the amount of substrate that you actually get to have that second phase. So just when you're trying to count carbons and figure out where everything goes, just kind of keep that in mind as we continue along. So yeah, so as we're talking about phase 1, this is again just kind of keep in mind which part's which. We're going to have that phosphorylation of glucose, and it's going to end with those 2 molecules of glyceraldehyde 3-phosphate, and we're going to use 2 molecules of ATP in phase 1. In phase 2, we're going to be taking those 2 molecules of glyceraldehyde 3-phosphate to pyruvate, And as we do that, we're actually going to get 4 molecules of ATP and 2 of NADH. So that is taking into consideration all of that doubling. So this is actually kind of your like total amounts. So this is a really nice slide. Just kind of look at like, all right, so what goes in, what comes out. So you put in 1 molecule of glucose. At the very end, we're going to get 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. And that's glycolysis. And one of the other things with this whole phase 1 and phase 2 is in phase 1, you're actually not going to have any oxidation. In the second phase, we will go ahead and have 1 oxidation step. So the other nice thing with this whole phase 1 and phase 2 breakdown is that phase 1 is actually the only part where we're using our ATP. Phase 2 is in kind of where we're getting all the good stuff. So it's nice, a lot of different parts of the breakdown that's useful. And here in this diagram, essentially what they're doing is they're showing that split. So this way, instead of trying to imagine like where did all of my carbons go, in this particular diagram, you just see the split, but what you'll notice is, again, you see everything that's duplicated, and let's just kind of keep track of where all of the carbons of our glucose molecule are. Where will our pyruvate go? So while we're going to spend a lot of time in glycolysis, there's going to be that question, what happens to the pyruvate after we're done? So pyruvate's going to have 3 main fates that we're going to concern ourselves with. The one that is so big that we're actually going to save it for after this coming up exam and spend more time on it later is going to be the TCA cycle. So TCA stands for tricarboxylic acid. So you're going to hear, we'll have some disclaimers about all the different names for that one. We'll worry about that one when it comes around. But that is one of the fates of glycolysis, or is one of the fates of pyruvate out of glycolysis. Pyruvate can also go through lactic acid fermentation. So this is what can happen in our muscles. We will be talking more about that later. Pyruvate can also undergo, under anaerobic conditions, fermentation to ethanol. This one's more common in yeast. Humans don't do this, but we'll also be spending a little bit of time talking about that later as well. Under normal circumstances, pyruvate, in animals at least, will get converted to acetyl coenzyme A, which then of course will go to TCA cycle and eventually produce carbon dioxide. When we're limited in oxygen, that's when we're going to use lactate, and alcoholic fermentation happens in yeast, also without oxygen. We're going to be talking a lot about coupled reactions. So this is something that we spent time on in the before time, and something that comes back a lot of times because the body often couples reactions to make things go. So these are also going to help us convert some of that metabolic energy of glucose into ATP. So we're actually going to see some substrate level phosphorylation as we go through. Now, only about 5% of the energy of glucose is actually released in glycolysis. And that sounds like a small amount, and it is. We're going to get a lot more from the electron transport chain. But 5% is also a significant amount. So it's not a lot, but it is still something that we want to keep track of and something that we do want to remember as we go along. And those coupled reactions, especially that ATP hydrolysis, is going to be what's used to drive the glycolytic pathway. So just like whenever we talked about coupled reactions before, typically the point of a coupled reaction is that you have something that you would like to go forward but isn't spontaneous on its own. It doesn't actually have, it would be unfavorable to go forward. So coupling that reaction is something that is favorable will allow it to move forward. And we're going to see that happen here in glycolysis.